Hi, in this video I'd like to go over a few things with you in Chapter 5 on membrane structure and function. Now, of course, in Chapter 4 we looked at cells and the cell organelles and the functions there, but this is going to focus really on the actual membrane and go into some detail about what the structure is like for a plasma membrane or cellular membrane, and then really take a look at transporting materials, substances, chemicals, ions, water, whatever the substances across the membrane, either in a passive format or an active format. So let's look at the plasma membrane first, its structure and function. As we've said before in Chapter 4, all cells are going to have some type of cellular membrane, this type of plasma membrane. It doesn't matter if you're looking at prokaryotic cells, eukaryotic cells, bacterial cells, animal cells, plant cells, they're all going to have a plasma membrane. That serves as a separation. It separates the internal cytoplasm, the inside of the cell, from whatever is on the outside of that particular cell in that particular environment. Sometimes it kind of compartmentalizes cells so that you may have certain cellular reactions that are not compatible, and so it kind of creates a compartment for certain cell reactions that take place. And we're going to be talking about this concept here, the fluid mosaic model of the cell membrane. That's a good vocab word to add to your notes because that's going to describe the structure of the plasma membrane. And that structure is basically a phospholipid bilayer. So let's look at that word for a minute. Phospholipid, we know from previous chapters that lipids, what a lipid is. And so, this is a nonpolar type compound. It's not soluble in water, and so this is a special type of a lipid that it has phosphate attached to it. And then it's a bilayer, just means it has two layers. And so let me show you the picture here in Figure 5.1. This is a characteristic classic view of a phospholipid bilayer that you would find in a plasma membrane. So you've got two layers of lipids here. All right, so a little structure there. That's my lipid. That's my chain of fatty acids. It has what's called a hydrophilic head. I'm kind of getting ahead of myself now, but we're going to talk about this in just a minute. But see a hydrophilic head on the outside. That's the water soluble section of it, and then the lipid soluble section of it, or the lipid based part of this membrane, is on the inside. That's the hydrophobic area here. And so you have two layers of this that kind of go back to back. And so this is why this is a phospholipid bilayer. And that's kind of what this is talking about. The external surface has hydrophilic polar heads that's soluble with water. And then the inner surface has the inner portion of the bilayer is hydrophobic. And since you do have two layers, the cytoplasmic surface is also hydrophilic. So again, we look at this diagram out here. If you see where the little cursor is, out here is the extracellular portion. And they'll label that extracellular matrix, or ECM. So you have exposure to that hydrophilic polar head here. Because you have two layers, you also have a hydrophilic head internally as well. So inside would be your cytoplasm inside the cell. Now, generally you find three main components. Of course, you're going to find the lipid, the phospholipid bilayer, but you also find proteins and you also find cholesterol. So those are the three major components you're going to find. We're going to spend some time talking about the proteins. Back to Figure 5.1, you see proteins that usually, most of the time, span the entire lipid bilayer, but not always. Sometimes they are, like, if it spans all the way across, it's called an integral protein. Basically, it's incorporated into the plasma membrane. Sometimes you see peripheral proteins, and so they're just kind of on the, in this case, kind of on this inner surface of the lipid bilayer. And you also find cholesterol molecules kind of scattered throughout. There's some little small lipid cholesterol molecules kind of scattered internally in that membrane. Please note that the cholesterol there is important. It allows for the fluidity of that membrane. In other words, that membrane is a fluid type membrane. So these protein molecules are free to kind of move around, kind of float around in this matrix here, or this bilayer. They're not fixed. In a fixed position, they can actually move around a little bit, and that creates some fluidity. And then also, if you notice in the drawing, how it's kind of curved around, and so that is kind of a mosaic model. The mosaic just means you have different molecules here. You've got some proteins here. You got some carbohydrates attached to it. You got the, of course, the lipid on the inside. You got cholesterol there. So you have a mosaic of components there. This is just a closer in view. Figure 5.2 kind of shows you that integral protein again. This is a protein that's embedded in that lipid bilayer, and it kind of shows you what one of those phospholipids would look like individually, with the hydrophilic on the outside, the hydrophobic on the inside. All right, as I mentioned, you mostly have proteins that are part of this, integral type proteins, but they usually have some attachments to them. Usually they're carbohydrate attachments, and so you generally see these words used, glycoproteins or glycolipids. And so because they use the prefix glyco, that refers to glucose or carbohydrate, some type of sugar type molecule chain or carbohydrate chain. Protein is the main root of the word, and so a glycoprotein is a protein molecule that has some carbohydrate attached to it. A glycolipid, again, is a lipid type molecule that has some carbohydrate chains attached to it. We'll talk about these a little bit more. Sometimes they help in cell identification. They kind of serve like as a name tag for the cell, if you want to think of it that way. They also they also kind of allow for some attachment of other molecules. Sometimes there's other molecules that, so these sometimes would serve as like a receptor type molecule as well. We'll see examples of that a little bit later on. So these integral proteins we mentioned a few times, there's some very important functions of these. We'll go through several examples here. Channel proteins, carrier proteins, cell recognition proteins. These are pretty easily named here. Channel proteins creates a passageway through the membrane. So we'll look at that. Carrier proteins also allows for a passageway, but it really is more specific as to which molecule that can go through that passage. And so I'll show you an example of that too. Cell recognition proteins. These are integral proteins that help to recognize foreign substances. So say, for example, your immune cells, your immune system could recognize something that's not supposed to be there, maybe a foreign cell, a bacterial cell, viral cell, or something like that. Receptor proteins, these are part of a cell signaling pathway, and so cells respond to signals from other cells, and this is like how your hormones work in your body. And so we'll see some examples of receptor type proteins. They receive information and then respond to some type of chemical signal, like a hormone would. Enzymatic proteins, some proteins, we've already talked about enzymes a little bit, so some proteins function as enzymes and help to speed up certain types of metabolic type reactions. So sometimes you have proteins that are associated with the cell membrane, either integral or peripheral based proteins that help serve as an enzyme. Then some proteins are junction proteins. That's because they help to attach adjacent cells together. So let's kind of take a little closer look and look at some examples. Channel proteins, probably the easiest here. Not that any of these are really that difficult, but this is probably the easiest one. You can see that you got the lipid bilayer shown here. I've got an integral protein that really serves as just a passageway and allows usually small molecules or small ions here to kind of pass through. Sometimes channel proteins can be gated, so they're only open. They've got like a little gate there, and they may open or close depending on some type of signal, or they could just be freely open like this and allow molecules to kind of pass in or out. So channel protein. A carrier protein does allow transport, the transport type protein, but it's usually very specific as to which molecule that it allows to go through. And so there's something known as sodium-potassium pump or sodium-potassium transport, and this usually requires, or a sodium-potassium transport requires energy to get this molecule or this protein to conform and allow that sodium ion or that potassium ion to kind of pass through. So this is not as simple as a channel protein. It's not always available. It's not, this is selective to certain molecules. This is how the example here is talking about how glucose can get into your cells. You have to have insulin in order to activate a carrier protein or open up that carrier protein to get the blood sugar out of your blood into the cells, like your muscle cells or liver cells. Cell recognition protein. Again, this is kind of like having a name tag on a cell. This is how your cells, if you think about your immune system, how your cells recognize what belongs in your body and what is supposed to be there. There's a huge concept or term up here, MHC, major histocompatibility complex. If you take some immune classes, you'll learn a lot more about these. But this is like an identification tag, glycoprotein. So it's a protein here with a carbohydrate attached to it, but it serves as a signal or really a identification, so that your white blood cells, your immune cells, know which cells to attack and know which cells not to attack. A receptor protein, you have lots of type of receptors on cells, and that means that they receive information. And again, a lot of this has to do with hormones. Hormones get secreted from endocrine cells, endocrine glands that go into the bloodstream. They find a target cell that has a particular receptor that matches that molecule, and they bind to that receptor, and they cause some kind of cellular response. Enzymatic proteins is what it sounds like. These are proteins that serve as enzymes. Enzymes speed up or catalyze certain types of reactions. And so this is just an enzyme that's kind of embedded in the lipid bilayer. And this is trying to show you some little molecules down here that go through a chemical type reaction because they bind to that enzyme, and that helps to speed up that particular reaction. And the last one here is a junction type protein. Junction proteins help to connect cells together. They form a junction, so that's kind of nicely named. And so what you're seeing up here in the diagram are two cells close together. So I've got two individual cells. Cell number one's up top, cell number two is down here. So they're very close together, but they're joined, held together by some type of junction protein. It keeps the cells very close together, and certain times during development or in cell growth, cells need to be really, really close together. All right, so that kind of gives you an overview of the plasma membrane. Kind of, again, don't forget about the fluid mosaic model and the main components of the plasma membrane and how those 6 main classes of proteins kind of work. How cells talk to one another, that's cell communication or cell signaling, and a lot of that goes into an endocrine type system. Cell receptors, usually protein type receptors, have the ability to bind to specific chemicals or specific chemical messengers, and that causes some type of cell response. And a lot of times it's referred to as a cascade type of response. And it kind of gives you an example down here that I've kind of already talked about a little bit. Let's say, for example, you eat a meal, and so now your blood sugar level increases, your blood glucose levels goes up, and so it goes up in the blood, and so the pancreas, this is an endocrine organ in animals, and so that releases a hormone called insulin that goes into the bloodstream. The blood that finds its target cells in, say, for example, the liver and also the muscle cells. That causes those cells to open up, use one of these transport proteins to open up and allow the blood glucose to be transported from the blood into those liver cells. or into the muscle cells where it's needed. All right, now we need to kind of change gears a little bit and talk about, now that we know a little bit about the plasma membrane, let's look at how it allows transport across the membrane. So there's a key word up here. You need to make sure you know this word, selectively permeable. You may have already heard this before. Sometimes you see this called semi-permeable. This just means that the plasma membrane has the ability to allow some molecules or substances or chemicals to go across the membrane, but it stops the movement of other molecules. Some things it doesn't allow to go through, or very easily. Usually, if the molecule is very small, the molecule is not charged. If the molecule is lipid soluble, it normally can go across the plasma membrane. It diffuses across that membrane. And they give you some examples up here. CO2, that's gas. Oxygen, that's a gas. They can go across the plasma membrane. Glycerol, that's a lipid soluble molecule. It can go across the plasma membrane. It has to follow what's called the concentration gradient, which is basically diffusion. And so that definition is there for you. Diffusion says molecules or substances or chemicals move from an area from a high concentration to an area of low concentration. There again, that's a good definition to know for diffusion, or establishing a concentration-based gradient, going from high to low concentration. Water can go across the plasma membrane, so cells can move water back and forth across. One term you may want to recognize here is called an aquaporin. This is a protein type channel that's really designed to help water go across the membrane. And so it's pretty nicely named. The aqua there references water, and porin we think of like a pore or an opening. And so water can move across the plasma membrane a little faster if it goes through the aquaporins. Usually if it's an ion, ions are charged particles, or charged atoms. And so if it's an ion, sodium, potassium, magnesium, calcium, or some kind of polar molecule, it's going to need some type of carrier protein. And so usually those carrier proteins are going to require some kind of energy source, ATP type energy. And so that's going to be referred to as active transport. Anytime you see active transport, that means it requires some type of energy. It's not going to happen just by diffusion alone. We're also going to look at a couple other definitions here. Bulk transport. Let's say you want to move large particles in and out of the cell. Your cells have to do that from time to time. Let's say you're secreting some type of substance. Maybe you've got, again, some kind of gland cell, maybe like an oil gland, or a sweat gland, or even some of your hormone type endocrine glands. They produce a substance that has to be secreted in bulk outside of the cell. They just can't diffuse that across the cell membrane in large quantities, and so there's a process known as exocytosis, and the cell is able to package what it's trying to secrete into a vesicle, and then that vesicle fuses with the plasma membrane, and it's like it dumps that, or allows that product or that secretion to exit the cell. The reverse is also true. So cells can take in large quantities at a time, and so it can essentially wrap its cell membrane around a particular substance, particle, and create a vesicle, and to basically engulf something. That's endocytosis. So this little diagram kind of does a nice job of showing some examples of, this is figure 5.4, showing some examples of transport. We've got our lipid bilayer. I've got some, notice that's going to be a channel protein that's embedded there. Got some other proteins. And so if it's just a charged ion, if it tries to diffuse directly across the membrane, it's not going to be able to go through the membrane again. Even though it's small, it has a charge associated with it, and so it doesn't want to just freely go through the membrane. Water can go through a channel, and evidently this would be, they didn't label it here, but this must be an aquaporin type protein channel because you've got water going through. If it's a non-charged type substance, then which would also be lipid soluble, it could diffuse directly across the plasma membrane. If it's a larger macromolecule, maybe something like glucose, pretty large molecule, it can't just diffuse across the membrane. So it would have to have, if it needed to come into the cell or go out of the cell, it would need, either some type of carrier protein, or it would need to go through endo or exocytosis. Nice little table here, table 5.1, that just kind of summarizes the type of movement. And it's a good table to kind of look at and study. Different types of movement are listed here, or passage across or transport across the membrane. Diffusion, the direction goes from high to low concentration. It requires that concentration gradient, and gives you some examples. We're going to talk about facilitated transport in just a minute. It also goes high to low concentration, but it's facilitated. In other words, it has to have help, and it has help through a channel or carrier type protein. Active transport, remember that requires energy. We said that, but one of the reasons it requires energy is because you're going the opposite direction of a concentration gradient. You're going toward higher concentration. You're going from low concentration to high concentration. That goes against diffusion, and so that requires energy to take place. And then bulk transport can go either inside or outside the cell. It does require some kind of vesicle to move that molecule in bulk. All right, section 2 talks about passive transport. Passive means it doesn't require energy. Remember that a solution is you take a solvent, you take some type of, usually a liquid is your solvent, and you dissolve something into that solvent. That's called a solute. You mix those together, and you have a solution. And so diffusion is whenever you take molecules and you move them down a concentration gradient. In other words, you're going from high concentration to low concentration. This is a very natural process. It's passive in that it doesn't require any kind of energy. Now, once the concentration on both sides of that selectively permeable membrane has been kind of equalized, then your net movement stops. You don't have, there is no more concentration gradient. There's not higher on one side and lower on the other. If they're about equal, they're pretty, you've reached an equilibrium, then your net movement stops. Diffusion can be affected by different things, such as temperature. Increased temperature, molecules speed up their motion, and diffusion is going to increase. Right? Pressure affects diffusion. If you have higher pressure, you can push molecules faster or easier across a membrane, that sort of thing. The size of the molecule, smaller molecules can diffuse faster than others. Figure 5.5 kind of gives you an illustration of just some simple diffusion to look at. Maybe you've done something like this before, perhaps not with the goldfish, but if you added a drop of food coloring in a glass of water, you can kind of see, you put the drop in there and it'll start to diffuse out. These molecules of dye or food coloring, whatever, start to diffuse out, and eventually they'll have an equilibrium gets met, and you have an equal distribution of those molecules in that solution. So that's diffusion. Now, osmosis is a special case. The reason it's special is because it deals with water. And so in this case, the water is moving. The water would be the solvent. And so the solvent is moving instead of some type of substance instead of the solute itself, instead of something like an ion or glucose or the dye or the food coloring. You're talking about water moving across a selectively permeable membrane. You can also define it as the diffusion of water. Okay. And so it's water going across a selectively permeable membrane. You still had to have that concentration gradient. And so on one side, the solute concentration, on one side of the membrane is high, but the water concentration is low. On the other side, you've got the reverse. On the other side, the solute concentration on the other side is low, but the water concentration is high. And so water, one way to think about osmosis is water always wants to go, because of osmosis, water always wants to go to the area with the higher solute concentration. That's what this is trying to say here. Net movement of water is toward the lower water side, which is going to be the side that has a higher amount of solute. Right, so that's important. Let me read that one more time. The net movement of water, the direction of water across the semi-permeable or selectively permeable membrane is always going to be toward the side that has the least amount of water, the lower water side, which is also the side that has a higher amount of concentration, the higher solute concentration. So a lot of times when you think of cells, at least in a sense of in maybe like the urinary system, and you're talking about the flow of water, you think of water moving toward where the sodium ions would be. So if your body holds on to more salt, your body has salt, you have a high salt diet, and you've got a lot of sodium in your diet, then water tends to follow that. Water goes toward that area. So in other words, you're going to retain more body fluids if you have a high sodium diet. Reason a lot of this is taking place, or osmosis is taking place, is because of what's known as osmotic pressure. Osmotic pressure is that pressure that's generated because of the concentration gradient here, and that water is trying to move toward the area that has a higher solute concentration that creates that osmotic pressure. Now, what this means for cells is you've got three terms up here. Let's look at the terms and then we'll look at the diagrams. Isotonic, iso, that prefix iso means equal, equal to. And so if you have an equal concentration on both sides of the membrane, there's no net movement of water, right? You're at an equilibrium. So if you look at an animal cell and a plant cell, that's how figure 5.8 is showing you. Animal cells across the top. This animal cell, they don't show you the solution that it's in. It would be kind of nice if they had like a little container here to show you the actual solution. But if you can imagine this animal cell being placed in an isotonic solution, that means the concentration of the solution and the concentration inside the animal cell would be the same. You have kind of a, water is free to kind of move in, water moves out. It's in an equilibrium state. There's no net gain of water. Same thing about a plant cell. There's no net movement of water. That's different than a hypotonic concentration. Hypo means less than, lower than, so the concentration of the solute in the solution is lower than the inside of the cell. So let's look at hypotonic. I take a cell, in this case an animal cell, I put it into a hypotonic solution. So the solution concentration, solute concentration outside of the cell is less than the inside of the cell. All right. Water wants to move toward the area that's more concentrated. So the area that has more solute concentration is the cell, because the solution around it is hypotonic. So that means water, because of osmosis, wants to move into the cell. Now, if this is an animal cell, that could mean that the cell gets too much water and it actually would burst. It would lyse. Okay. Plant cells survive fine like this, and actually they need a little bit of this pressure. And so that's, in plants, it's called turgor pressure. And so here the cell walls kind of help support that and keep that plant cell from bursting. And so, same thing is going on. You've got water going into the plant cell in a hypotonic solution. A hypertonic solution is just the opposite. Hypertonic means, hyper means higher than or greater than, so concentration of the solute is higher in the solution than inside the cell. And so here in the animal cell, you put it into a hypertonic solution, there's more solute concentration outside the cell than there is inside. Water wants to go from inside the cell toward the area that has more solute concentration, which is now outside the cell. So that means water is trying to leave that animal cell. Too much water leaves, it causes the cell to shrink or to shrivel. That has a special vocab word called crenation. For plants, the same things are going to happen. If they're in a hypotonic solution, that'll happen. And essentially the plant will, what we would normally call wilt, it shrinks and actually can hurt the plant in a condition known as plasmolysis, because the plasma membrane actually, the cytoplasm actually shrinks and the plasma membrane kind of separates from that cell wall. All right, let's look at facilitated transport. I mentioned this briefly because of the table earlier. Movement of molecules cannot pass directly through the membrane lipids. We talked about that. Some molecules require some type of carrier. As long as the carrier does not require energy, then it is referred to as facilitated transport. The molecule, if it, number one, requires a carrier and that carrier doesn't require energy, then it's facilitated transport. You're still going from high to low concentration. That's why no energy is required. So this is kind of showing you some facilitated processes here. I got more solute on the inside here. On the outside, sorry. There's my lipid bilayer. This is inside the cell. This is outside the cell. Got more molecules here. And so they're going to want to go from high concentration to low concentration, but they're too big just to go through the cell membrane. So they need a carrier protein to help that process. And so they can go through facilitated transport through that carrier molecule, carrier protein without any energy requirement. Active transport. Anytime you see active transport, automatically think of energy, ATP, energy. Movement of molecules. And the reason is because they're going against the concentration gradient. They're going from low concentration to high concentration. That's just against diffusion. That's not an easy task to complete unless you have energy to help accomplish that. And because and it also is going to require some kind of carrier protein as well. Classic biological example of an active transport system that is found in living organisms is what's called the sodium-potassium pump. It uses energy in the form of ATP to move sodium ions out of the cells and move potassium ions into the cells, to kind of store them up, and that is going against their natural concentration gradient. All right, so let's talk about briefly exocytosis and endocytosis. These are just great vocabulary words to study. You'll see some of these on the next module exam. They're pretty easy to know. Again, you're dealing with larger type of molecules that need to be transported in or out of the cell. And so exo is exiting the cell, exocytosis. Endocytosis is coming into the cell. It involves, both of these processes involve some kind of storage vesicle. So if it's exocytosis, the storage vesicle has a molecule inside of it. It fuses with the membrane and secretes that content, that substance outside the cell. Endocytosis is the reverse. The cell, in fact, engulfs the substance into a pouch-like vesicle or pouch-like structure that becomes a vesicle, and that takes it into the cell. If you look at specific examples, if you're talking about a solid material, you're talking about a solid substance here, not a liquid, then it's called phagocytosis. Phagocytosis. Literally, this means cell eating. Phagos is eating. And so a good example, your immune system, you got white blood cells, their job is to try to engulf, take in particles, maybe bacteria, maybe some kind of damaged cell debris, maybe even some kind of virus infected cell, to take it in, destroy it. Pinocytosis, pino refers to fluid, and so this is literally cell drinking. If there's some kind of liquid substance or liquid molecules that the cell needs to take in. Sometimes the pinocytosis uses receptors to get this process started. And so if that's the case, if there's a receptor that some kind of protein that's getting a signal to get this process to work or to begin, then it's referred to as receptor mediated endocytosis. This little diagram here is just kind of showing you some examples here. I've got my plasma membrane. This is exocytosis. I've got some kind of storage vesicle. You got a molecule inside of it. It fuses with the plasma membrane and it releases whatever that substance may be, some type of perhaps a chemical, or like your sweat glands or oil glands in the skin. This is also showing the same type of thing. Here is this is endocytosis. This is a phagocytosis, cell eating. And so notice how that plasma membrane kind of wraps around. And eventually just continue wrapping around it to form that pouch type structure, and it takes in the molecule into a vesicle. Pinocytosis, same type of thing, except you got some type of liquid or very small solute ion or something. Receptor mediated also works similar, but in this case you've got some type of receptor molecule that has received a basically a signal to get this to work.